Well, thank you very much, and it's good to be back at the ISNTD. Also delighted to see Gabriella's talk, and it's very illuminating because, you know, we have to think like a mosquito, we have to think like a snail, think like a tsetse, but very few of us will be able to think like a snail. The aquatic habitat is very different, and that's where basically schistosomiasis plays out. And as Marianne says, we often have a schism in malacology, which is the study of snails which are involved in medical diseases, because technically they aren't vectors, they're intermediate hosts. The more attuned to being innocent bystanders, because they don't really seek us out, they're just part of the natural environment. And it's actually the larval stages of the parasites that have the host-seeking behavior, which you see in the other vectors. So I'd like to talk a bit about work we're doing at Liverpool School in Cameroon. This is part of the Countdown network, which is looking at interventions which are appropriate for the scale-up of NTDs. One of those is schistosomiasis. I have my colleague here, Louise Hamill, who's also doing some vector control work on blackfire with regard to onchocerciasis. So the story unfolds out in Cameroon. Cameroon has a large problem of schistosomiasis, but we're going to be looking in the southwest, in the Crater Lakes. So I'd like to quickly introduce to you the tools we have for snail control. They're quite blunderbuss approaches, and you'll see the model of why there's a blunderbuss approach, but also how our theory of snail control is a little bit weak in comparison to what we have for other vectors. I'll set that into active studies going on in Cameroon, how we're trying to think about targeting the snail control interventions, and also give you a bit how we're thinking more broadly, how we want to shape future policies, making sure that we don't forget snail control is an important part of the control for schistosomiasis, especially when we start to think about elimination of that disease. This is a nice graphic. It just shows you the life cycle of the disease schistosomiasis. So the key components are infected people. That's not working. Infected people who can be treated, so we have a lot of emphasis on providing access to praziquantel for school-aged children or other age groups who are of school age who would like to receive treatment. But I just want to point out, not everybody gets treatment. You've got people who are vulnerable, adults, and also young children who aren't serviced in treatment campaigns. So there's always a weakness there. People's behavior spread the disease, so when we think of mosquito and tsetse finding behavior, we spend a lot of time in schistosomiasis trying to understand human behavior and why people have indiscriminate defecation or urination. So that's a challenge to try and put strategies and correct it, because we have water and sanitation. Also, when you think of the aquatic environment, it's necessary for agriculture, so there's a strong link not only with water development for hydroelectricity, but also for food production, so that also impacts on schistosomiasis. And then here we have the aspect of snail control. I want to remind you that aquatic environments like schistosomiasis have a very fluid life cycle. So this is static, but the reality is it's a very complex mashup of people's behaviors, the density of people, the number of parasites coming out, and how we deliver control options. Nonetheless, snail control was originally pioneered in the late 60s, 70s, but it started to come back into view again, because if we want to contain environmental transmission, we have to dampen the snail's role. And then there's a new manual coming out by WHO, refocusing the tools of the trade for schistosomiasis control. So we'll go into this. Now, our only tool for snail control basically is formulations of the chemical molluscicide niclosamide. And there has been a change because it's now produced at low cost in China, several manufacturers, and that also brings in another story. China are very keen to support schistosomiasis control in Africa because they've had good inroads into schistosomiasis control in China, also using snail control. So the Chinese have been supporting bilateral agreements with several countries in Africa, including Cameroon, trying to export some of their knowledge and practices into the African setting. Now, some of that is appropriate. So these are one of the manufacturers making low-cost niclosamide, but it boils down to this. This is our kind of tools cabinet for schistosomiasis control. We have the human diagnostics, we have some of this fecal sampling, we've got urine dipsticks, but we now have in the cupboard some snail control chemicals, which are slightly different formulations to what used to be available. But This is the big problem, and it probably goes what Gabrielle was saying. You have to know the animal that you're trying to locate as well as kill. Now, the Chinese snails that transmit schistosomiasis are very different to the African ones, and also the way in which you might use molluscicides in China is very different to what you might use in Africa as well. So keep that in the back of your mind. So this is where we are with snail control. We have aquatic habitats which fringe areas where people are living. This happens to be a fishing village on Lake Victoria on an island. Consider the aquatic environment very deep, very extensively colonized by snails, but those snails can constitute a population which might be considered to be a haystack, a dry haystack. So the idea is, if there's an infection event, so imagine if you strike a match, that environment might be very receptive for the transmission of the disease, if those snails are susceptible. So you strike the match analogy is a single person or a handful of people urinating or defecating into that environment, and that's sufficient, often alone, or it might be multiple events where you've got more matches being struck, to generate infections, and that has two outcomes. You either get infection taking off in the snail, and one snail being infected can have a massive repercussion for subsequent transmission, or you're striking it onto a damp haystack where the infection doesn't take off. So I'm just pointing out that if the environment isn't manipulated to favour that damp haystack, you're always going to have the possibility of having schistosomiasis transmission. A few individuals can maintain that transmission. So if you don't have the snails, and you don't have the haystack, then you can't have a problem. But the problem is, how long can you keep snail populations at bay, and how much does it cost? And that's the problem with snail control. So the advice WHO have, you have to approach snail control very carefully. Planning stage, a pilot intervention stage, and then you go to full implementation, and then you conduct monitoring and evaluation. And for those of you unfamiliar with some of the snails, this is biont filaria, found in Africa, transmits Mansoni. Globosus transmits hematobium in Africa, but the snail in China, Onca melania, very different to those two. It's an amphibious snail, it's not an aquatic. So, activities, snail monitoring, snail identification, characterization of habitats. Aquatic habitats are very complicated. They have a lot of heterogeneities there, and it's difficult to find snails at times in a quantitative manner. Application of molluscicide can be Hudson backpack sprays, which many vector people who give out insecticides will know quite well. It can be adapted for use of niclosamide, large-scale pumps, giving it out to large-scale areas. So those are the tools of the trade, but now we put it into Cameroon. This is where we're looking at two crater lakes down the southwest of Cameroon called Mbo and Kotto, and we've been taking a staggered approach where we're looking at the disease, how people are interacting with the water, and also trying to think what is the best way to conduct snail control. So just highlight a paper that we recently came out as part of the group. Two crater lakes, about 20 kilometers away from each other, quite different actually. This is Kotto, this is Mbo. Mbo's in a protected area, so it's in a forest area, but there's a focal point where you find snails, and that's probably the transmission point for that village. In Kotto, there's an island in the centre, and there's a lot of daily water contact, as you see. Now, using chemotherapy, we can get the disease down to fairly manageable levels at Mbo. It's almost pushing it towards elimination, but at Kotto, we're still at about 50% prevalence. So we think about intensifying treatment and also expanding treatment and trying to handshake that with other control mechanisms such as snail control. So here we have a child in the water probably being exposed, but hopefully he would have had treatment either once or twice in the time he's been at school. At Mbo also there's been large-scale interventions onto water and sanitation approaches. So putting snail control, you can't think of snail control by itself. You have to put it into the epidemiology of the disease, and in so doing, people who are studying the epidemiology of the disease have to know how to target the snails themselves. So we had community mobilization, sensitization, discussions of what's going on. We've provided more access to treatment. It's got the disease down to about 15-20%. But this is where we start to think about targeting that snail control even further. This is the perimeter of the lake at Kotto. This is the island where most people are living on. We find an interesting snail, Balinus cameroonensis, that's the transmitter. We also find another snail which has been introduced probably from Asia, Indoplenormis, which has no role in transmission whatsoever, but we can use that as a marker snail of infections. I just want to point out, when you do the snail surveys, you can map out habitats. You can also find infected snails that are shedding cercari. But we also want to know a bit more about the ecology of those snails, but also the ways in which people use the water themselves. So I just want to point out that if you go back a year later, the Balinus population is more or less the same size as you would expect. The Indoplenormis must be going through a little bit of a colonization succession because it's increasing quite significantly in numbers. So this is where you think about how do you target the snail control. We've been using GPS data loggers to look at the water contact patterns of people, in particular mums and children. We can map it out on the island to show where these water contact sites are. I think it would be evident to say you cannot use molluscicides around the perimeter of the lake, it's too expensive. Could you use it around the perimeter of the island? Probably not. You need to target where you're going to deliver it. So we have that information now to say where you have the most likely water contact types, you try to use a molluscicide to target it. And I just want to point out, I know I'm short of time, rather than looking at the interesting history behind the Indoplenormis, when you use a molluscicide, we've got some key questions. It kills fish, so fishing is important in that lake, so you have to think of ways to balance the ecological impact against the benefit you have for snail control. Microgeographic coverage, where are you going to apply it, how long are you going to do it, and knowing that, if you only have incomplete coverage, you're always going to get recolonization of that habitat by snails, so it's like holding the populations at check. So, done preliminary experiments with some formulations of nicolosamide as wettable powder as well as specific concentrate. Interesting, Indoplenormis is not as sensitive as the Balinus to the molluscicide. So what we're thinking of now is trying to target that by using slow-release formulations, which you get in swimming pools. If you put a chlorinated tablet, it can disperse through just drip feed. So we're thinking of perhaps using chained dispersal plastic pots effectively put out into the water and used remotely to kind of give that anti-snail effect, but also anti-cercarial and miracidal action too. So I think that's just saying why you need to target and also be mindful of the ecology. And I just want to draw attention to a couple of articles we've been writing to think about that, putting it into a strategy for long-scale control. And then just thanks to everyone in Cameroon, thanks to ISNTD and friends and colleagues in Liverpool and Cameroon. So thank you.